Most of the breakthroughs come out of labs where people cheat the system, or they have private donors, or they have some way to do it. Then the successful scientists basically get their grants to do what they've already done, and then they can use the new money to do something new, and that's how they make breakthroughs. But you have to cheat the system a little bit to do that.
So when we first started reading the genetic code, we call it digitizing life. It's converting the 4-letter code into ones and zeros in the computer. And we did this first in 1995 with the very first genome. And there are fundamental questions of, does DNA contain all the necessary information for life? So we decided to go the other way and start with the ones and zeros and see if we could recapitulate life by doing that. So we used some of the information from the first genome, but we had to learn how to actually write the genetic code. We did this first with a small virus that infects E. coli. And the virus was about 5,000 base pairs long. We made it chemically. We injected it into E. coli. And this was the first experiment of what happened. E. coli read this synthetic piece of DNA, like normal DNA, started making proteins. The proteins self-assembled and made the virus, and the virus killed the cells. So we call this a situation where the software builds its own hardware. We wrote a piece of chemical code, put it in a bacteria, and it made a virus. So our goal wasn't to make a virus, it was to make an entire cell, one base pair at a time. This took much longer than we thought it would, but over that time, we developed a wide range of technologies that enabled us to actually write the genetic code very effectively. So the chemistry was one side, the biology was another. And we had to develop a way to boot up now this total synthetic chromosome that we had made.